Before we get started, it's important to note that the Merriam-Webster dictionary defines the plural form of the word octopus as both octopuses and octopi.
Recently, The Atlantic published an article titled, "Octopus Do Something Really Strange to Their Genes." In this video, we will discuss how cephalopods are able to change their RNA through RNA editing. But first, let's introduce our star.
The phylum Mollusca includes a large number of primarily aquatic invertebrates. Within that phylum is the class Cephalopoda, which includes octopuses, squids, cuttlefish, and nautiluses. These creatures are known for a wide variety of incredible adaptations, such as their advanced intellect, ability to fit into small spaces, and ability to change color. Next, you'll learn about yet another amazing adaptation, their ability to change their RNA.
Let's first get started by looking at the central dogma of biology. The central dogma of biology is a thematic concept that governs microbiology. It describes the process by which DNA becomes RNA, which becomes protein. DNA, the tiny molecules that holds all of our genetic information in our cells, is made up of just three parts: a deoxyribose sugar, a phosphate group, and nitrogenous bases. These simple parts make up one of the most important molecules in our bodies. DNA is like a foreign language to ribosomes, which produces proteins in our body. Thus, the DNA must undergo a process called transcription, in which the DNA is converted to RNA. This RNA is more of the ribosome's language, but it must undergo further processing known as translations. The RNA molecules are translated by ribosomes into triplet codon sequences and produce strands of amino acids which form proteins. Now let's look at how DNA is edited in humans.
DNA editing is done by a variety of nucleases. These nucleases work by breaking the DNA on both strands and combining them in a new way to create a mutation. A common and well-known method of DNA editing is known as CRISPR. In this method, the Cas9 enzyme cuts out a section of DNA and replaces it. It does this by causing a double-stranded break in the DNA, which is then repaired by homology-directed repair to cause a mutation. Homology-directed repair, also known as HDR, uses a homologous template of the DNA to repair the DNA and is considered flawless. Originally a method of defense against viruses for bacteria, CRISPR now has the potential to treat disease or knock out or end various traits.
Now let's take a look at RNA editing.
RNA editing is conducted by the enzyme ADAR, where discrete changes to the nucleotide sequences within an RNA molecule occur. This could include insertion, deletion, and base substitution of one or more nucleotides within the edited RNA molecule. ADAR encodes the enzyme responsible for RNA editing by site-specific deamination of adenosine. This enzyme destabilizes double-stranded RNA through conversion of adenosine to inosine. In human RNA editing, the intron on the pre-messenger RNA are mutated, resulting in mutated pre-messenger RNA. This has no effect on the protein produced, though, because the introns are spliced out in messenger RNA processing, and only the exons are used to code for the protein. RNA editing is extremely rare in human beings as it compares to other species like cephalopods. Only 3% of human genes undergo RNA editing. Most of that 3% occurs in the introns that will get spliced out through RNA processing and eventually degraded, therefore having no effect on translation of RNA to protein.
Unlike humans, cephalopods can achieve up to 57,000 RNA edits. Most of these edits happen in the exons of the RNA, which are the nucleotide sequence that is translated into proteins. Their ability to edit their RNA allows them to adapt much more quickly by changing the protein sequences versus years of evolution. RNA editing is much more common in the cephalopods with complex brains called coiloids versus their prehistoric and simple brain, Nautilus.
Many of the sites edited by coleoids are shared within their subclass.
To wrap up this discovery, there are many further implications to this. In the study of how large nervous systems are put together, there are still many questions. For example, why do cephalopods do RNA editing, whereas it is very rare in humans? Why do other species not edit their RNA to the same extent that cephalopods do? The next experiment the scientists plan to conduct is research on ways to genetically manipulate cephalopods and their DNA. Through this, they could disable their ADAR enzymes, stopping them from editing their RNA, and to see what happens with that.